Hi everyone, and welcome to today's Biology Daily Booster. This is number 7 in our Paper 2 series. It is also an advanced information listed topic on both the higher and foundation biology, so hopefully we're going to find some relevant information in today's booster for us all. What we're going to be looking at this point are some of the key scientists and what they've contributed to our understanding of genetics. And if there is only one person that you remember as far as genetics goes, then I would suggest it is this guy, Gregory Mendel. Now, Mendel is known as the father of genetics because he was pretty much the first guy to really do extensive work into this. What he did was he actually spent a lot of time with peas. I know it might not sound thrilling, but what he actually did with those pea experiments gave us this first massive insight into the world of genetics. And what he did was, through his observations of his peas, he found certain characteristics were passed on from parents to the offspring. So from that, he determined that characteristics in plants are determined by these things he called hereditary units, things that are passed on from parents to offspring. And these units are passed on one from each parent, so both parents contribute here. He then went on to say that those units are either dominant or recessive. The other guys that also contributed, but perhaps aren't as well known, we start off with Friedrich Miesha. Now, he discovered a stuff called nuclein. That's an acidic substance present in the nucleus. We then come to Avery, and he actually transferred genes between bacteria. And what he saw there was the ability to cause disease can be transferred from one strain of bacteria to another. And then those bacteria pass that ability on to their offspring. So from that, we showed that genes were made of DNA. Next guy, Chargoff, he discovered base pairs. So this is one where they don't necessarily come out with a question asking you who discovered base pairs, but his work can be phrased as a question and has been in the past. Now, he discovered that even though different organisms have different amounts of DNA, all DNA, no matter what organism it came from, had equal quantities of adenine and thymine and equal quantities of cytosine to guanine. So this is where a base pairing question comes up. So they could give you something like, in an organism, and they could pick any random one, 23% of all of the bases were adenine. And then ask you how much would be thymine. So because adenine is 23, we would also have 23% as thymine. They've got to be the same because of complementary base pairing. If they're being a bit trickier, they could have actually said, how much was cytosine? We'd only know the adenine, so 23% there, but because we know adenine, we know thymine is also 23%. So 23 and 23 gives us 46%. Then 100 minus 46 gives us 54% left, which is then going to be split between cytosine and guanine. So that means that we would have 27% cytosine and 27% guanine. So it's just a maths-based question. We know it's got to add up to 100. We know that adenine and thymine have to be the same. We know cytosine and guanine have to be the same. So whatever they give you, you can work out any of the others at all. We then come on to 4 people whose work kind of very closely interlinks. First ones, Franklin and Wilkins. Now, Rosalind Franklin was the one that actually photographed the DNA crystals using X-rays. It was her photographed image that Wilkins passed on to Watson and Crick that allowed them to come up with this double helix structure of DNA that we know today. If Franklin hadn't taken that X-ray image, Watson and Crick wouldn't have come up with that structure. So this is where we've got scientists working together to actually come up and further science. Now, what we have with that double helix, remember, are 2 spirals held together by complementary base pairs, and between those complementary base pairs, we've got hydrogen bonds. So those base pairs, A's, T's, C's and G's, held together by hydrogen bonds. Since then, we've continued working on genetics. It wasn't a case of we now know it's a double helix, job done, let's all just sit and have a cup of tea. Instead, we've looked at it to see what else can we do here. So since then, we've discovered individual genes, particularly ones that code for inherited disorders. We've developed an entire new field of genetic engineering, which we will look at more detail in a future booster. So there's been ongoing research in these areas, and in some cases, the research we're now doing is only possible because of advances in technology. And whenever we've made a discovery, what we have to do is publish the work that we have got. A couple of reasons. First, by publishing the work, it allows for this idea of peer review. Now, when we talk peer review, that's where other scientists are going to read your work and they try and replicate it. Assuming you have done things all above board and you're a good scientist, then other scientists will be able to replicate your work. And that obviously adds support to it, and then it makes whatever your findings were much more valid. If, however, you're a little bit ropey and a dodgy scientist, when people try to replicate your work, it's not going to happen. And this has happened in the past with a few different examples. And at that point, we then just can completely debunk it and say, well, this isn't actually the case. So peer review is important for that reason. Publishing your work is also important because it allows other people to know what's been actually discovered and then build on it with their own work. So it's not a case of every single lab around the world has to do that same bit of work themselves before they can build on it. Through sharing that knowledge, we can then just move on to the next stage together. One of the major developments in the recent past was in 2003 when we completed this thing called the Human Genome Project. The Human Genome Project was a global collaboration to try to sequence all of the genes in the human body, because the genome, remember, is the entire genetic material of an organism. So what we were trying to do was sit there with the entire human genome, all of the genetic material in humans, and then put together a sequence of the whole thing. And we did it. In 2003, we successfully completed the human genome. Now, and to allow us to do that, we've had some advances in technology that made it a much quicker process than how we used to do it. And since that point, we've now been able to look into the field of gene therapy, which again we'll look at in more detail in a future booster. But the basics of it is we can now identify where the faulty genes are, and we can develop ways to replace them with normal copies of that particular gene so that it's going to work properly as it should do. The Human Genome Project was one of the kind of most important things that we've done in the recent past, because that's now opened up these new branches of genetics to us. Do remember though, this is one of those examples of where scientists across the world have worked together. It's an example of where we've seen technology improving to allow us to do this faster than we expected. And as technologies continue to improve, then obviously it's become a little bit more accessible to more people. These days you can literally order a tube off the internet, spit in it, send it off to a lab somewhere, and get a basic evaluation of your genome. So you can identify if you've got certain markers for genetic conditions. They'll tell you how much DNA you've got that came from various countries around the world, etc. So as technology improves, prices come down, becomes more accessible to all as well. If there are any bits on today's booster that you want to go over in more detail, obviously there's the videos on the main channel you can use. I would also recommend using your revision guides, just going through, test yourself, go over some past exam papers, do some of those questions just to make sure you really know this stuff. And of course, don't forget to join us again tomorrow for our next biology booster.